The QuickKO Human Gene Knockout Kit is a ready-to-use product with proven guide RNA knockout effect. A comprehensive assessment of target genes and cells through documentation and trial tests is recommended before knockout experiment. Let's take the example of the liposome transfection method and adherent cells in 6-well cell culture plates. First, cell preparation. Seed the cells in the logarithmic growth phase into a 6-well cell culture plate 1 day in advance to make sure the cell fusion rate maintains 60% to 80% during transfection. The experimental group is used to transfect QuickKO guide RNA and optimized SpCas9, while the control group doesn't transfect any plasmids. The NC guide RNA in the kit is a guide RNA sequence from unrecognized human origin or known murine DNA, which can construct a negative control cell line according to demands. Second, cell transfection. Blend QuickKO guide RNA and optimized SpCas9 in a 1:2 ratio, and transfect target cells according to the parameters of trial tests for transfection efficiency. Typically, the total amount of DNA required for 1 well of the plate is about 2.5 microgram. Third, hybrid clone screening. QuickKO guide RNA vector possesses 2 screening labels: puromycin resistance and red fluorescence. Either resistance screening or flow sorting can be selected to concentrate transfected positive cells. Here we would like to introduce the puromycin resistance screening in detail. First, compound the screening medium containing purinomycin with complete medium according to the lowest lethal concentration of the drug confirmed by trial tests. Then replace the previous medium with the screening medium after 48 hours transfection. So do the control group. Next, renew the screening medium every 2 to 3 days until all cells of the control group are killed. Finally, transfer the experimental group cells back into the complete medium without drugs, and continue to culture for 2 to 3 days for hybrid clone cells. Fourth, hybrid clone identification. Hybrid clonal identification includes sample processing, PCR amplification, gel electrophoresis, and Sanger sequencing. Sample processing. Take a portion of hybrid clone cells, almost the 4th power of 10, into a PCR tube, centrifuge them at 1000 RPM for 5 minutes, then discard the supernatant. Compound buffer A and buffer B in a 50:1 ratio into 1+ cell lysis buffer. Then add 20 microliter of 1+ cell lysis buffer to the cell sample and mix evenly. Incubate the reagent at 55 degrees Celsius for 20 minutes, then inactivate it at 95 degrees Celsius for 5 minutes. Centrifuge the lysate product at 12,000 RPM for 5 minutes. And transfer its supernatant into a new PCR tube for later use. PCR amplification. The kit involves nested PCR and requires 2 rounds of amplification. The template in the first round is cell lysis supernatant with primers F1 and R1, operated in 0.5 degrees centigrade touchdown PCR procedure. The template in the second round is the product of the first one with primers F2 and R2, operated in a conventional PCR procedure. Note that the enzyme should be placed on ice to dissolve completely and mix evenly before use. PCR reaction system preparation should also be done on ice. Set positive and negative control group with control template and water correspondingly. Gel electrophoresis. Take 5 microliter of the second round PCR product and do electrophoresis detection with a 1.5% agarose gel under a constant voltage of 160 volt for 20 to 30 minutes. The bands of the experimental group should not be larger than that of the positive control group. Negative control group should be free of amplified bands. If multiple bands occur, the strips of various sizes can be recycled and purified separately for Sanger sequencing. Sanger sequencing. Do the Sanger sequencing on the PCR products and compare the results with wild-type gene sequences. If there is a set of peaks or base deletion at the knockout site or region, monoclonal screening will be continued. Fifth, monoclonal screening. Monoclonal cells can be obtained by single-cell sorting equipment such as a flow cytometer, or by limiting dilution method. Here we introduce the simpler and more convenient dilution method. Digest and centrifuge the mixed clones, discard the supernatant, and resuspend them in complete medium, then count them. Dilute cells to a concentration of 8 to 10 cells per milliliter with complete medium. And seed them in 96 well cell culture plates at the amount of 100 microliter per well. Culture the cells in an incubator with 5% carbon dioxide at 37 degrees Celsius for 2 to 3 weeks. During this period, observe regularly under a microscope. Mark the wells in which monoclonal cell colony grow, and properly replace or supplement the medium. 6th, monoclonal identification. Digest the monoclonal cell colonies in separated wells respectively and turn them into single-cell suspensions. Take 1/3 of the cells and carry out single clone identification in the same method as the mixed clone identification. Transfer the remaining cells into 24 well or 48 well cell culture plates for amplification. The Quick KO Human Gene Knockout Kit targets coding genes and adopts a multi-guide RNA strategy. It can generate 2 types of gene knockouts including indels and fragment deletions, and will preferentially select single clones with fragment deletions for Sanger sequencing. The following circumstances usually occur in monoclonal identification. 1. The sequencing result shows a single peak. 3 cases may appear through further contrast with the wild-type gene sequence. A. Result is completely consistent with the wild-type genotype. B. The number of indels is a multiple of 3. C. The number of indels isn't a multiple of 3. The third case is generally considered as a successful knockout. 2. The sequencing result shows a set of peaks. Now the PCR product needs to be cloned by TA and then do the Sanger sequencing, or directly the third-generation sequencing. Successful knockout of monoclonal cells should ensure that all genotypes produce indels whose quantity aren't multiples of 3. That's for the instructions of Quick KO Human Gene Knockout Kit. For more information, please scan the QR code on the screen or visit Nest website. Retrieve information in this section, enter the gene name or ID, quickly locate your desired kit.